In this lesson, we're going to be diving into the phylum Porifera, which includes the sponges. And this is a picture of me actually with a purple vase sponge. And I think this was my trip to Mexico, if I recall correctly. It is a Caribbean sponge, wherever that might be. All right, so to recap where we left off, we saw how the phylogenetic tree branch for animals can be further divided into different animal groups, including the sponges, which are the first animals to have thought to have evolved in the evolutionary timeline. So, being that they are referred to as the basal group of animals, which is the first animal group to diverge from all the others. Sponges are thought to be monophyletic, meaning that all sponges share a common, most recent common ancestor with all the other sponges. So this is the hypothesis that your book presents. However, as with all taxonomic studies, there are different hypotheses out there. Some suggest they're paraphyletic, but we're going to go with the monophyletic hypothesis. Okay, so having all the sponges coming from a single most recent common ancestor, they would have certain characteristics in common with each other. These are the characteristics that define the sponges, or the phylum Porifera. Most of them are marine, however, there are some freshwater sponges. They are all sessile, meaning that they don't move on their own. They're kind of just stuck wherever they are, wherever they settle, um, wherever the larva settle and start growing, that's where they remain for their duration of their life. Now, sponges have an asymmetrical body plan, so there's no line of symmetry. They have a very irregular shape. They also come in a variety of sizes and colors, but they're very beautifully colored, though, underwater. With a flash, you can see a beautiful orange sponge on the left and the purple sponge like I was photographed with on the right here. Now, unlike other groups of animals, sponges do not have tissues. Although they lack tissues, though, they are composed of different types of cells, and that's what we're going to be talking about next. So, while we're talking about their mode of obtaining food or nutrition, you'll see some of those examples of different types of cells, because basically sponges are filter feeding particles of food out of the water. That's how they get their nutrition. They capture particles that are suspended in the water as they pass through their body. So they do this by drawing water in through their pores into a central cavity called the spongocyl, as shown on the diagram at the bottom right here. And then the water gets pumped out through an opening at the top called the osculum. So the water is always flowing in that direction as shown by the blue arrows, in through the pore to the spongocyl and then out through the osculum at the top. Now, some folks have figured out that you can actually watch the movement of the water through into the sponge and up out the osculum by putting a fluorescent dye next to the sponge. So there's a video if you'd like to watch this. I'll put the link on D2L as a shortcut, but it's also embedded here in the PowerPoint. Okay, now there's additional cells that we haven't yet mentioned, and you might actually recognize some of these cells that are lining the interior surface of the sponge. And those are the choanocytes. They kind of look like choanoflagellates, don't they? They have that collar cell with a flagella. So these are the cells that are responsible for generating the current. The flagellum actually move just like for a choanoflagellate, except instead of allowing the cell to actually take like move around outside of the sponge, it's stuck there, embedded in the wall of the sponge, the sponge's body. But as those flagellum beat, it generates the current that allows the water to enter the pores and then get pumped through the spongocyl and out the osculum. Sponges also have a specialized cell called an amoebocyte, might sound familiar. We talked about like amoeboid movements when we learned about the protists. So it's kind of similar to that. Um, they move using pseudopodia like an amoeba. So that's like an extension of the cytoplasm is how those amoebocytes move. It's this blue structure here that's right next to the choanocyte. So what these amoebocytes do is they actually absorb the food that's taken in by the choanocyte through phagocytosis. Then it gets absorbed from the surroundings, including from the choanocytes, and then transported to other parts of the sponge body. So that's why the amoebocytes have to be able to move so that the materials, the food, can get to other parts of the body to maintain the health of the sponge. So I just want to go back to actually I have it here at the top, just as a reference of where these things are located. Remember where the choanocytes, or sorry, yeah, the choanocytes are here on the inner lining of the sponge. And then right next to it, this is like a zoomed in of this guy here down at the bottom. But notice how the amoebocytes aren't always right next to the choanocyte. Some of them have moved away. Perhaps they're on their way to bring that food to other parts of the sponge, or they're on their way back to get more food and then transport that. So be sure that you understand what makes the sponges unique from other animals, what are those general characteristics of the sponges, as well as these specialized types of cells that allow the sponges to get their nutrition. Now for fun, maybe you didn't see this before, maybe you did, but in the picture I showed earlier of the orange sponge, there's actually a hidden fish here. He's very well camouflaged. Can you see it? If you don't want to have a spoiler and you're still looking, pause the video now because I'm about to show you where it is. There he is right in the middle. That's a frogfish. It's a type of anglerfish, and they use biomimicry to camouflage within their host sponge. So I actually took this picture, and the other sponge I just recalled that was actually taken in Bonaire, which is an island in the Caribbean. And I had never seen a frogfish like this before. He was down in a crevice in the sponge, and our dive guide actually agitated him a little bit to get him to come out, which I wasn't too keen on, but I was glad to be able to get a picture so I could incorporate that into the lesson and share that with you all. So there's lots of cool examples of biomimicry in the world, including those animals that live in or on sponges. Sponges are like their own little microenvironment. All the nooks and crannies inside are really a great habitat for lots of little creatures to live in and have protection, and possibly even get some food from the sponge as it's filtering through and putting water out the osculum. Okay, so that's where we leave it for the sponges.